Hello and welcome to our program titled Ushering in a New Era in the Treatment of Chronic Kidney Disease and Anemia, Appreciating the Patient Burden, Exploring Pathways and Pathogenesis, Reflecting on New and Emerging Data. I'm Dr. Ajay Singh, Senior Associate Dean for Postgraduate Medical Education and Director of the Master in Medical Sciences in Clinical Investigation Program at Harvard Medical School. I'm also a nephrologist at the Brigham and Women's Hospital here in Boston. I'm joined today by my esteemed colleagues, Dr. Edgar Lerma, Clinical Professor of Medicine from the University of Illinois in Chicago, Illinois, and by Dr. Jay Wish, Professor of Clinical Medicine at the Indiana School of Medicine. Dr. Wish serves as Chief Medical Officer for outpatient dialysis at Indiana University Health in Indianapolis, Indiana. The faculty disclosures are displayed on the screen for your convenience. This program is supported by an educational grant from GSK. We would like to thank them for support of this initiative. Our program today will include a robust presentation and panel discussion on new therapies that are in development targeting HIF-PHIs. The learning objective for this initiative is as follows. Cite the efficacy and safety data of novel agents used to manage anemia in CKD. I'll be providing a brief overview, and then Dr. Wish and Dr. Lerma will join me for a panel discussion. The current HIF-PHIs in development are shown in this next slide. On the left-hand side, you see a list of these agents that have already been discussed briefly by Dr. Wish. Roxadustat, vadadustat, daprodustat, molidustat, inaridustat, and desidustat. The ones circled in green, Roxa, Vada, and Dapro, as Dr. Wish mentioned, have undergone global clinical trial development and are being evaluated by the US FDA. Molidustat and inaridustat are under development in Japan, and desidustat is under development in India. On the right-hand side, you see the different phases that these agents have undergone, and you can see that roxadustat and vadadustat have not received FDA approval, and it's likely that additional studies will be needed. But they are approved in other countries, including Japan, China, and in Europe, depending on the agent we are speaking to. Daprodustat has undergone and completed phase 3, but is now currently being reviewed by the FDA, and further information will become available as the FDA completes its review. Daprodustat has been approved in Japan. Molidustat and inaridustat are not undergoing development in the United States and are approved in Japan. Desidustat is under development in India, has undergone phase 2 trials there, and has been approved in India for use. The next slide depicts a build around different HIF-PHIs and their efficacy and safety. Let's start with roxadustat. This agent has been found in studies to be superior to placebo in non-dialysis patients and to ESAs, erythropoiesis-stimulating agents, in dialysis-dependent patients in both increasing hemoglobin and decreasing hepcidin. In post-hoc analyses, thromboembolic events in non-dialysis and dialysis-dependent patients with CKD have been analyzed by the US FDA. And safety analysis have shown strong dose-dependent relationships in humans, but there's been no effect that has been reported on platelet production, activation, and thrombosis in healthy and 5/6 nephrectomized mice. Vadadustat was shown to be non-inferior to conventional ESA, both for non-dialysis and dialysis patients, for both increasing hemoglobin and decreasing hepcidin. With respect to MESA outcomes, vadadustat was found to be non-inferior to conventional ESA in dialysis-dependent CKD patients. However, with respect to MESA outcomes in non-dialysis CKD, vadadustat increased MACE outcomes by 17%, which was statistically significant. But as shown in their New England Journal paper, this seemed to be restricted to the non-US population rather than the US population. Daprodustat was reported in New England Journal papers to be non-inferior to ESAs in non-dialysis and dialysis patients for both increasing hemoglobin and decreasing hepcidin. In both the non-dialysis and the dialysis population, daprodustat was non-inferior also for CV safety in these populations. There was, however, in the on-treatment analysis, MACE and tumor adverse effects, which now appear to be methodological flaws in those analyses. Moving to the next slide, here you see that there are disparate results from HIF-PHI studies. And the question really is this because of differences in study design and study conduct, or could the differences be attributable to differences in biochemistry and pharmacological characteristics? Depicted below, you see in the three boxes the data for roxadustat, vadadustat, and daprodustat. And you see that there is a different dosing schedule that is used for each of these three agents. So they have differing potency in humans. The schedule is different because their half-life differs. For roxadustat, it's 3 times a week because it's got a longer half-life, whereas for vadadustat and daprodustat, the schedule can be once a day or 3 times a week. Both have been demonstrated to work. The half-life is shown below that and reflects this dosing schedule. Metabolism also seems to differ. Roxadustat and daprodustat have a similar metabolism with interaction with the CYP, cytochrome P, C8 system that is similar between roxadustat and daprodustat. The data on vadadustat isn't available at the present time. With respect to relative activity, roxadustat is thought to impact prolyl hydroxylate domains 1, 2, and 3 equally. Vadadustat and daprodustat have relative activity for PhD3 more than PhD1, and then more for PhD2 in that order. On the next slide, what you see is the situation currently with respect to HIF-PHIs and FDA for roxadustat and vadadustat, which I'll cover first, and then I'll add in the information for daprodustat. For roxadustat, on August 12, 2021, the FDA issued a complete response letter, known colloquially as a CRL, in declining approval. The FDA cited, quote, concerning safety results, untested proposed mitigation dosing strategy with unknown efficacy, sicker patients with greater need to demonstrate safety, and difficulty in obtaining more definitive data in the post-marketing setting, unquote. For vadadustat, on April 1, 2022, the FDA also issued a CRL declining approval. And here the FDA stated, quote, failure to meet non-inferiority in MACE in the non-dialysis patient population, increased risk of thromboembolic events driven by vascular access thrombosis in dialysis patients, and the risk of drug-induced liver injury, unquote. On April 19, 2022, the FDA accepted GSK's application for daprodustat, and the PDUFA date has been set for February 1, 2023. The application is based on data from the ASCEND phase 3 clinical program, and in both non-dialysis and dialysis CKD patients, daprodustat helped improve or maintain hemoglobin levels, and in the intention to treat analysis, there was no increase in MACE outcome when it was compared to events in patients treated with conventional ESA. So getting to the PHI overview in this next slide, you see depicted are advantages on the left and disadvantages on the right. With respect to advantages, there's evidence in humans that's been demonstrated and reported that there's improved iron utilization with HIF-PHIs, reduced triglyceride, total cholesterol, and low-density lipoprotein levels, and reduced peak EPO levels. Of course, Dr. Wish has already alluded to the fact that the oral efficacy could be a major factor in convenience of dosing and access to the drug. Additional potential benefits could include, although this has not been demonstrated in any of the trials published so far, a potential for reducing mortality, protection from ischemic injuries, and effective in patients resistant to ESAs. So there's some evidence to support it, whereas additional potential benefits have not been demonstrated in trials so far. On the right-hand side, you see the disadvantages. There is evidence for a predisposition to pulmonary arterial hypertension, although none of the trials have definitively spoken to this potential adverse event. Additional potential harms, again, with no data so far: tumor progression, thromboembolic disease. There's some concerns expressed for the ROXA program and for the VADA program with respect to thromboembolic disease, but that was in a post-hoc analysis. Cyst growing promoting effects in patients with autosomal dominant polycystic kidney disease, or ADPKD. Again, not been demonstrated in any of the trials so far. Pro-angiogenic effects in patients with vascular retinopathy, such as diabetic retinopathy, also not demonstrated as yet, and then vascular calcification. So a lot of potential disadvantages, but very few, if any, that have been demonstrated so far in the phase 3 programs. So key takeaways. HIF-PHIs have reached the final stages of regulatory approval. They have demonstrated efficacy, improving or maintaining hemoglobin levels, improving iron utilization in some studies, but not in all, for some agents, but not in all, and likewise, reducing cholesterol levels in some studies, but not in all. Approval has been denied for both Roxa and VADA. With respect to Roxa, the FDA cited a thrombosis risk, and VADA, in addition to the thrombosis risk, the FDA cited the MACE adverse event that was observed in the non-dialysis population. The daprodustat application was accepted by the FDA and is pending with a PDUFA date for February 1, 2023. There have been disparate outcomes that have been reported in the published literature with all of these agents, which could result from methodological differences in trials or differences in patient populations, or in fact, pharmacological differences between these drugs. There are important advantages and disadvantages to consider should HIF-PHIs become available.
Thanks, Dr. Singh. So I'd like our panelists to address the issue of whether the safety signals that were observed with roxadustat and vadadustat and were the result of the complete response letters from the FDA denying approval, at least for the time being. Is this a class effect, and should we expect it to apply to all the HIF-PHIs, or are there in fact pharmacological differences among these agents that perhaps would predict that maybe daprodustat would not have some of these issues and perhaps could be potentially approved by the FDA? Dr. Singh?
Of course, all the HIF-PHIs are potent prolyl hydroxylase domain inhibitors and that are orally bioavailable small molecules. And they have a common moiety structurally. They all are potent inhibitors of the three PHD domains. They're mostly metabolized by the liver. So there are many similarities. And they are very similar in correcting the hemoglobin. However, there were differences between these agents in their trial programs in terms of the types of patients that were recruited, the geographic location of where these patients were sourced. On the pharmacological differences, I think it is important to recognize that these small molecules do differ structurally, and they do have different half-lives, and they do have different impacts in terms of drug-drug interaction. So I think my view is that while it is tempting to say the predicate is that all these three agents work in the same way, and so therefore the adverse events from one agent can be generalized to the whole class, I would argue that the data doesn't quite support that, in that, as the FDA has done so far, they've taken an individualized approach to reviewing each of the agents and deciding whether they're going to move them forward in the approval process.
Well, let me ask you a follow-up question on that. Do you think the FDA, for instance, was tainted by the results from roxadustat, and that may have contributed to the vadadustat disapproval?
Right. Dr. Wish, you know, and I know that we can't get in the minds of our FDA colleagues. So we're not, quite sure what might have influenced their decision-making. What we do know is VADA itself did have a problem, right? And in the New England Journal paper, they themselves reported that there was a MACE signal that meant that it failed the non-inferiority criteria. In fact, it was inferior to conventional ESA. And I think that really did influence the FDA's perspective over on top of whatever their concerns were with Roxa. So I sort of leave this to them to figure out, because they have access to the complete study results from each of the programs to judge whether they're ready for moving forward on the approval process.
So let's move on about some of these potential disadvantages of the HIF-PHIs that Dr. Singh mentioned in his presentation. You said that a lot of these are theoretical concerns. Are there any of these actual concerns that have been demonstrated in clinical trials or even anecdotally in case reports? Dr. Singh?
Yeah, so for Roxa, for example, in the New England Journal papers that they published with respect to the Chinese patients, they did note hyperkalemia. While these agents generally seem to be quite well tolerated, there was this MACE signal that was seen in the non-dialysis population that granted Dr. Lerma's points well taken. We need further information to sort of tease out what exactly is going on there. Because I can't imagine that a drug that you're exposing the population, population around the world recognizes the passport of that person. So it seems to me that, either is possible, right? It could be that safety signal wasn't detected in the US population, or in fact, that the safety signal in the non-US population was influenced by some quirk in the study design or study conduct. And for DAPRO, of course, there was concern that was reported in the on-treatment analyses for both a higher rate of MACE and a higher rate of tumor-related morbidity mortality, which, as we explained in the New England Journal, may be related to how events were counted, particularly in those patients randomized to darbepoetin, and that this may be a dosing interval bias. There have been concerns reported from preclinical, when you use very high doses preclinically with respect to VEGF levels, and then in the Shuvash Russian population, these patients develop polycythemia and potentially pulmonary hypertension, but those have not really seen the light of day in the clinical trials. Now, it is true to be very cautious. Clinical trials select out a population. They're not real-world evidence when you've used these drugs in millions of patients. So we do need to be cautious in not making overarchingly broad statements about safety. And I don't think that the FDA did do that. And I think, with any drug, whether it's HIF-PHIs or anything else, the jury's always out until you've actually used the drug in clinical practice.
Yeah, I agree with that. But the Japanese and Pacific Society of Nephrology, and as you point out, 5 of these 6 agents have been approved in Japan, have a position paper that in fact does recommend pharmacovigilance with regards to some of these potential adverse events. But again, none of these have been demonstrated in clinical trials. What about the cholesterol and iron effects of HIF-PHIs? The cholesterol lowering effect was mentioned. Is this something that has been seen with all the HIF-PHIs? And then the iron effects, we talked about the mechanism of action in terms of improving iron absorption and increasing transferrin levels and decreasing hepcidin. Are they being seen with all the HIF-PHIs, or does this seem to be more specific to some agents or others? Or are perhaps the sub-analyses of the large-scale clinical trials ahead for perhaps roxadustat that reported these clinical trials earlier and had the luxury of the time to do some of these analyses?
Well, I think
Dr.
the answer
Singh?
is you nailed both, right? I think it's quite possible that there may be differences between these agents in terms of their action and their potential benefit with respect to cholesterol. Of course, the cholesterol LDL benefit would be huge if it was then translated into causality with respect to cardiovascular complications. The iron benefits seem to be mostly related right now to biochemical benefits on transferrin and hepcidin. It's very hard, I have to be frank with you, to start seeing differences in iron utilization because these trials, A, were not designed to look at iron utilization, and second, it's so difficult to monitor and quantitate iron benefits because patients receive iron in so many different ways, either on dialysis or if they're not on dialysis. But it is very promising to see the iron benefits from a metabolic standpoint. And from what we know, if you impact hepcidin, it should translate into differences in absorption in iron and iron metabolism. Thank you again, my colleagues, for this very spirited discussion. We've reached the end of our allotted time for today. I'd like to thank Dr. Lerma and Dr. Wish and everyone for participating and listening in on the HIF-PHIs in development segment of this series. I'd also like to thank GSK for their support for this program. We would like to ask learners to please follow the link that is provided at the end of the program and in the thread below to fill out this evaluation and post-test in order to receive CME credit. This provides us with valuable feedback that will be used for the planning of upcoming programs. This is part 3 of a 4-part series. There will be one additional social media broadcast on October 5th, 2022 at 2:30 p.m. Eastern Daylight Time. Please follow the Iridium CE social media Twitter or Facebook page so you will be sure not to miss updates. Thank you again for joining us.